All right, so what we've talked about until now is taking our image, let's say we have an image of the brain, and making this image sensitive to diffusion in any direction. If, on the other hand, we generate an image where we are sensitive to diffusion only along a single direction, what will that image look like? Well, there will be attenuation of signal for anything moving along this left-right dimension. So in this image, for example, the corpus callosum will have lower signal amplitude than, let's say, white matter bundles like the superior longitudinal fasciculus that run front to back in the head. If you do that, why is that the case? Right? The reason that is the case is that in white matter, and I deliberately chose these two white matter regions, if we think about the anatomy, we basically have many structures that look like this. We have bundles of axons which are all running in a similar, within some constrained distance, are running along a single direction. And all of those axons, whether it's their membranes, the myelin surrounding them, the neurofilaments and microtubules inside the axons, whatever it happens to be, we have this setup of all of these parallel-oriented structures. All of that structure, those membranes, that's the stuff that I was talking about in tissue, which water molecules are going to run into and have trouble transgressing. So when we talked about the simple example of our bucket of water, or my glass of water sitting here on the table, where diffusion could move in any direction, in tissue, we said that there was all kinds of other stuff, macromolecules, cell membranes, organelles, that slowed down the rate of diffusion. But because there was a relatively, I don't want to say random, but because there isn't a sort of systematic organization of this structure, those water molecules are limited in the direction that they can move, and pretty much in any direction that they move, there are going to be limitations on that motion. In white matter in the brain, because of the unique structure of white matter, the barriers to diffusion are set up so that in this example, if we look at the likelihood of some population of water molecules and the direction they're going to move, right, they will have a very hard time moving across or perpendicular to the orientation of those axons or that fiber bundle, however you want to describe it, simply because of the orientation of all of these structural components. Whereas along the long axis of the axon, or of the fiber bundle, there are relatively fewer barriers to diffusion. That doesn't mean that there are none, right? But there is going to be a preferential movement of water molecules along this long axis of the fiber bundle. That's why if I generate an image over here, which is sensitized to diffusion in one direction only, left to right, the orientation of the fibers, corpus callosum fibers moving left to right in the brain. There is a large amount of diffusion along the length of those fibers and relatively little diffusion perpendicular to those fibers. So in this example, this is a brain region where there is a dramatic amount of diffusion moving left to right. In fact, the rate of diffusion left to right in the corpus callosum here is going to be much greater than the rate of diffusion, let's say, in gray matter in the cortex, which has a less systematically organized structure. And in gray matter, we have an inhibition or restriction of diffusion in pretty much all directions. On the other hand, In this example, which I called the superior longitudinal fasciculus, which is a white matter bundle that runs front to back in the head, so these axons are lined up anterior to posterior. There is a large amount of diffusion in this direction and relatively little diffusion left to right. So in the presence of a diffusion sensitizing gradient left to right, there will be very little attenuation of the signal at this location. And in this image, we will see higher signal in the region of the SLF than we will in the corpus callosum. Is that clear? So looking specifically at white matter, if we sensitize to diffusion in a single linear direction, we are going to differentially affect the signal based on the orientation of the white matter fibers. And if, for example, there would be some obliquely oriented fiber bundle, we will detect an attenuation proportional to the amount of diffusion, the vector of diffusion that's parallel to that fiber. Okay? Question? Yeah.
Will you be able to get velocity from these images?
Sure. This is a diffusion-weighted image like any other. The only difference is that to acquire this image, we only turned on one direction. So in this example, we would have, I should qualify it, assuming we have a B0 image to which we can compare this, we can make the same quantification of velocity as we do with our other diffusion-weighted image. And that's exactly what we'll do. From this, we will derive an ADC image, which is telling us about, in each voxel, the no longer the speed, but the velocity of diffusion at each voxel, or the component of velocity parallel to this diffusion sensitizing gradient. Is that clear why that's the case? Yes? Any questions about this? So from this one image, we can tell something about the orientation of diffusion. Now, it turns out that if we acquire a series of images, and let's say we sensitize these images to diffusion, each in a different direction. In this case, each in an orthogonal direction. So in image number 1, where the diffusion sensitization is on the x dimension, image number 2, the diffusion sensitization is in the y dimension, and this one is z coming out of the board. So if I asked you to indicate on this image on the left, where would we have the highest signal amplitude? Where would we, on this diffusion-weighted image, sensitized left to right, where would we have the highest signal? We would have it along areas where there was diffusion perpendicular to the direction we're sensitizing. And we would have the lowest signal amplitude in areas where diffusion was along that direction. In this example, would look like this. Okay. And if we looked at one more area, let's say the internal capsule, where diffusion is moving in and out, right, perpendicular to the slice. So what would diffusion in the internal capsule look like if we sensitized on a left to right direction? It would be relatively high signal. Right? What about over here? Same thing. And in this image, where would we have our lowest signal? The diffusion going in and out of the slice would be most attenuated because that's the direction that we're sensitizing. whereas the other two structures that we're looking at would have relatively high signal. So I think you can see that there is a pattern across these three different images for each location based on the three-dimensional trajectory of diffusion. Now it turns out that if we have, that if we have enough of these images, meaning multiple images that are each sensitized to diffusion along a unique direction. So we might have an additional image, for example, which is sensitized along the XZ direction. Okay. And we could have many, many images, each sensitized in a different direction. So if we have a sufficient number of these images, and the minimum number is 6 unique directions along which we sensitize to diffusion, we can actually take each of those 6 images, which are diffusion-weighted images, in comparison to the B0 image, generate an ADC image for each of these, a quantitative diffusion-weighted image, and use that velocity of diffusion under each of these conditions to compute for each voxel what the 3D trajectory of diffusion is like in that location. Okay. And this is something called the diffusion tensor. We can do potentially an even better job the more different directions along which we sensitize to diffusion. But keep in mind that each additional direction requires a complete iteration of the entire process. Now, this is echoplanar imaging, so they don't take that long to acquire. But the more unique directions along which we sensitize to diffusion, the longer it's going to take us to do that. Now, we're making these measurements at a voxel in the tissue, which contains many protons. I don't mean to say, right, that in the corpus callosum, every single one of those protons is moving in exactly the same direction. First of all, when we looked at this example, this is not an absolute. It doesn't mean that in white matter, diffusion is only parallel to the direction of the fibers and never is perpendicular. It just means that it is much more likely for those spins to move along the axis of the fiber. But there is always a distribution. If there were a certain population of spins in this white matter bundle, there will be a distribution of directions of diffusion, such that some will be perpendicular, some will be parallel, and some will be somewhere in between. The preponderance in normal white matter will be along the axis of diffusion. But this range of directions of diffusion actually can be represented, right, as a 3D ellipse, which is, right, this picture over here, right, is computed from a single location in one of these diffusion-weighted data sets. So this three-dimensional ellipse, these are the XYZ coordinates in the image. So from a single voxel, we can actually calculate what the diffusion in that voxel looks like. And this has several characteristics. It's a long, skinny object, and its long axis is telling us the overall dominant direction of diffusion at that voxel. Okay? So I'll talk about this example in a second, but if we go back to our diagram, in the corpus callosum, the dominant direction of diffusion is left to right. So the orientation of this diffusion ellipsoid would be left to right. But even within the voxel that this represents, there is some range of directions of diffusion. That's reflected by how thick or how fat that diffusion ellipsoid is. In the corpus callosum, where diffusion is very uniform, where we have our most densely packed axons in the brain, this diffusion ellipsoid becomes extremely long and skinny because diffusion is almost all along that single direction. If we would look at the other extreme, use our same diffusion tensor approach to make a measurement from ventricular cerebrospinal fluid, what would it look like? It would be a sphere, because in the CSF there is no structure at all to limit diffusion in any direction. This will begin to approximate a sphere because there is no dominant direction of diffusion at all. In gray matter, for example, where we don't have this linear organization, this will also be relatively spherical. So the ways that we can characterize this diffusion ellipsoid is its principal direction of diffusion, something that is sometimes called the principal eigenvector, which tells us the overall direction from this voxel. This measure is not for the whole image or the whole brain, it's for a single voxel. And then there are two additional vectors which tell us about the girth of that diffusion ellipsoid. So the principal eigenvector, the longitudinal vector, is telling us the orientation of diffusion, and the girth of the ellipsoid is telling us something about how homogeneous the directional diffusion is at this location. So in a very highly organized white matter structure, such as the corpus callosum, we will have a very long and thin diffusion ellipsoid. That girth or thickness of the diffusion ellipsoid is expressed as something called the fractional anisotropy. This term anisotropy refers to the homogeneity of the direction of diffusion. So in cerebrospinal fluid, for example, diffusion can go in any direction because there's no structure to inhibit it. That's called isotropic diffusion. In corpus callosum, on the other hand, our diffusion ellipsoid is very long and skinny, reflecting the fact that diffusion is predominantly along a single direction. It doesn't go in those other directions. That's called anisotropic diffusion. The fractional anisotropy is simply a way of expressing how non-uniform the diffusion is. In normal white matter, the fractional anisotropy should be very high, which corresponds to a long and skinny diffusion ellipsoid. In gray matter, the fractional anisotropy is much smaller. The diffusion ellipsoid is more approximating a spherical appearance. So if we look at an image down here, this image is the result of computing this diffusion ellipsoid for every single voxel in this example in the slice. And the color image on your left is encoded so that the color tells us the direction of diffusion. It's simply showing us the principal diffusion direction. Right? Red is left to right, green is front to back, and blue is in and out of the plane of the slice. And I've blown up this area in the corpus callosum to show you that for each and every voxel, we can actually plot this vector. Right? So we're seeing the orientations of these vectors, and their hue on the color scale changes based on their orientation. This information, this directional image, is derived only from that principal diffusion direction, or principal eigenvector. Now, the anisotropy, which has to do with how skinny or fat this diffusion ellipsoid is, gives us a different type of information. It tells us how uniform the diffusion orientation is at that location, which can be useful because in the setting of tissue injury, let's say we're looking at a patient's brain and there is an area where there's a multiple sclerosis demyelinating plaque. So think about what happens in the presence of demyelination. When we looked at our diagram here, the reason that diffusion dominated along the long axis of this fiber was because of all of that linearly oriented structure, including myelin. In the absence of myelin, we can have a situation where these axons are spaced apart. The the myelin is gone. This is just one potential scenario. All of this space is areas where there may be diffusion that defies this linear orientation. Right? So this appearance would give us a much broader spectrum of directions of diffusion. This would be a lower fractional anisotropy. That diffusion ellipsoid would become much shorter and fatter because of the increased components of diffusion that are not along the direction of the path of the axons. So this image over here is an example of how you can color code based on the principal diffusion direction and do very detailed anatomic mapping of white matter pathways, simply based on identifying the principal diffusion direction. Any questions about this? You should know that this approach, which is diffusion tensor imaging, this is like a field unto itself. We're just trying to get some conceptual sense of what this is about. Now, we can also do things like this. Is my movie going? My movie's not going to work. Okay, so I'll show you the movie in a little while. But in this picture, right, this patient with a tumor, we have actually done essentially an in vivo anatomic dissection of his white matter. All of these color-coded little noodles that you see coursing through the image are white matter pathways. How do you do that? Well, if we think about this principal diffusion direction information, right? For each and every voxel, we can compute the diffusion ellipsoid and determine the orientation of the overall diffusion at that location. If we think about an image and we look at multiple locations, let's say we start out in the corpus callosum, which has fibers that run from left to right. So if we start out in this location and say, what is the principal diffusion direction? It's left to right. And then ask the question, if we look at a bunch of surrounding locations, surrounding voxels, and let's look at the orientation of diffusion at each of those adjacent locations. Do we find that there is a streamline or end-to-end association of the orientation of diffusion at our initial spot, which we could call the seed location, and those adjacent? And make that go in a stepwise fashion, find that across some trajectory there would be a continuous pathway of this principal diffusion direction. Right? And that allows us to reconstruct the location and orientation of diffusion doing what we refer to as, right, diffusion fiber tractography. And there are multiple different approaches to doing this, but just to conceptually understand what is involved is starting at one point and looking at the rest of the brain to try and find a continuous pathway where the principal directions of diffusion line up. Okay, I just want to show you one more example. Which is how we can use an assessment of the anisotropy, right, or how skinny, right, or fat that diffusion ellipsoid is to be very sensitive to the presence of white matter pathology. So this is an example. This is a patient who is a cocaine user who had some problems with performance on certain neuropsychological tests that correlate with frontal lobe function. So here is his typical structural image, right, FLAIR images. And if you look in the right frontal lobe, You don't see very much going on there. But looking at these two images, one which is showing us the principal direction of diffusion, color-coded as we saw it before, you can see that there is a region, unlike the other side, where we don't see the directional diffusion in the same way as we do in the unaffected part of the brain. And this image is a grayscale image of anisotropy, showing you that in normal white matter, such as the corpus callosum, internal capsules, we have very high fractional anisotropy, very low in the ventricles. So we expect normal white matter to have this very high fractional anisotropy. And you can actually see that there is a hole in the fractional anisotropy map in this guy, right, resulting from whatever the white matter pathology happens to be. Okay, so again, this is done by choosing a few different locations in the brain and asking the question, what's the principal diffusion direction at this starting location, which we can call a seed location, and where does that diffusion go from this point? Okay, so it's a 3D volume, and for example, you can see fibers in the corpus callosum or part of the corticospinal tract as it relates to the tumor. And it's a complete 3D model that you can turn around and understand the relationship of the anatomy to structure. So as I was asked before, you know, this is cool, but why would it make a difference? So the major clinical application of this at this point is to look at patients with brain neoplasms that are going to have surgery and try and help the surgeon make a more informed decision about how they're going to approach and deal with the lesion to avoid violating some of these major white matter pathways that could leave the patient with a deficit.
Are these individual axons that we're seeing, or?
No, these are not individual axons. This is all, by the way, you have to realize, is based on an assumption, which we discussed, which is that water is going to preferentially diffuse along the direction of the fiber. It's not actually showing you the anatomy of an axon or a fiber bundle per se. It's just showing you the direction of diffusion. Right? So there are, you know, the major advantage of this technique is that you can do this in vivo. There are other techniques which are much more reliable for tracing white matter pathways. But you can't do those in humans because the subject has to be killed to get the information. So while there are limitations here, and we've really just like barely scratched the surface of how to use diffusion tensor imaging, there are, you know, significant questions and potential limitations, but that's kind of balanced and tempered by the huge advantage of being able to do this in a living patient.